OHSU and PNNL. And today I'm going to start out introducing Peter Barr-Gillespie, who's our OHSU speaker, and then Bobby Jo Webb-Robertson will introduce Ying, who's the PNNL speaker. So Peter Barr-Gillespie almost needs no introduction for the OHSU side of the audience, but for the PNNL folks, Peter's the Executive Vice President and Chief Research Officer at OHSU. He's a professor with the Oregon Hearing Research Center and an affiliated scientist with the Volm Institute. He's been on the faculty here since 1999, but his connections here actually go back to the 80s when he did a senior undergraduate thesis at OHSU while finishing a chemistry major at Reed College. He did his PhD at UW in Seattle and then postdoctoral work with Jim Hudspeth at UCSF and UT Southwestern. And he joined the faculty at Johns Hopkins and was there for a number of years before coming back to OHSU. Peter's research focus is understanding the mechanisms that enable our sense of hearing, and particularly interested in the role of hair cells in the inner ear, and we'll hear about one aspect of that today.
Thanks, Beth. I will introduce Dr. Ying Zhu. Ying is a bioanalytical chemist in the Environmental Molecular Sciences Laboratory at PNNL. He did his PhD at Zhejiang University in China before he came to PNNL. And for the PNNLers, he also doesn't need much of an introduction. He is our nanopots guy. But for those at OHSU, Dr. Zhu has been focusing his research on the development of ultra-sensitive microfluidics and mass spectrometry technologies to enable the measurement of small amounts of biological samples. He leads the research directions of single-cell and spatial proteomics at PNNL, and he is the co-inventor of the nanopots technologies that you will hear about, and he has made significant contributions in applying nanopots in various biomedical research. So I have read some of the research papers in this area, but I'm really looking forward to the presentation. And so I will hand it over to Peter and Ying.
Thanks, Bobby Jo, and thanks, Beth. I appreciate the introduction. I'm sure Ying does as well. So I'm going to kick things off, and then we're going to switch to Ying, and then we're going to come back to me, and then we're going to go back to Ying. So we'll see if we make this work smoothly. If I could have the first slide. So the work we're going to talk about today, at least the biology part of the work today, is as Beth alluded to, we're going to be looking at the sensory cells in the inner ear, the so-called hair cells, which are responsible for our senses of sound and head position balance. And without going into gory detail about the anatomy of the ear, you can see here in this diagram that the external part of the ear, the ear, the ear on the outside, the pinna, and the ear canal lead into the middle ear and then finally the inner ear. And the inner ear is where the sensory cells live. Next slide, please. So in the cochlea, which I didn't point out, but you might have seen there before, cochlea is a complex organ with several fluid-filled chambers and different cells located at different spots within it. If you take a cross-section through the cochlea, like in the scanning electron micrograph, you can see part of the cochlea is made up of the so-called organ of Corti, which is a complex organ built out of 20 or 25 different cell types, including inner and outer hair cells, which are not pointed out here, but they're hair bundles. The protrusion on the top of the hair cell is pointed out. And the hair bundles detect the sound that comes into the inner ear and transmit sound information back to the brain. Next slide. So this is another scanning electron micrograph showing hair cells and their hair bundles. There's a fracture that allowed us to see the cells underneath the epithelium down at the bottom. And you can see how each of the hair cells has a hair bundle on its apical surface. And if we look at the hair bundle in more detail in the next slide, you can see on the left are some scanning electron micrographs of different auditory hair bundles. Far left with the turtle, and the two upper ones on the right from the rat cochlea, the two cell types, the outer hair cells and the inner hair cells shown there. And just for fun, bat inner hair cells are shown at the bottom right, and you can see that they are shorter, but with roughly the same features. I mean, if you compare all these different hair bundles, remember one hair bundle per hair cell, you can see that they have these individual stereocilia, which are actin-filled in the diagram there on the right, and they're arranged in a staircase pattern with short stereocilia giving way to taller and taller stereocilia. And movement of the hair bundle in the direction of the tallest stereocilia opens ion channels called transduction channels that depolarize the cell and influence neurotransmitter release. So we're really, in my lab, we're really interested in characterizing both the composition and function of the hair bundle, the stereocilia, but also we're interested in differences between outer and inner hair cells, for example. Next slide, please. So the problem with working, particularly doing protein analysis of the inner ear and hair cells in particular, is that there are only a few thousand hair cells per organ. Hair bundles, the hair bundle makes up just a few percent of the total protein in a hair cell. And the proteins we're most interested in, like the mechanotransduction proteins, are present at very low levels in hair bundles. So in all experiments characterizing the proteins of hair cells and hair bundles, we're pushing the limits of sensitivity with whatever technique that we're using. And back maybe more than 15 years ago, we decided to use mass spectrometry to start characterizing first the protein composition of hair bundles. And those early experiments used hair bundles from chicken because their auditory organ had about 10,000 hair cells, relatively easily dissected, and we could isolate the hair bundles. Next slide. So this method, the method for isolating the hair bundles is illustrated here, and we use this for prepping those samples for mass spectrometry. We basically dissect these vestibular organs, which have, like I say, about 10,000 hair cells lying flat on epithelium, and glue them into a little chamber. And we spread warm agarose onto the organs and let it solidify. And then we shear off the bundles by twisting the disc of agarose and flipping it upside down, and we have all the hair bundles left in the agarose, and the cell bodies and the rest of the epithelium is left behind on the cover slip of the chamber. And then we can excise out little blocks of agarose that have hair bundles in them to do our mass spectrometry experiments. And a before and after image of what happens is shown there on the right in those scanning electron micrographs. You can see how the bundles are sheared off at their bases. And the next slide is characterizes by immunofluorescence a little piece of the block of a block of agarose. The block of agarose is stained with phalloidin to label actin and DAPI to label nuclei. And each of those green dots is a single hair bundle with about 100 stereocilia. And you can see that the isolation technique works pretty well, probably gets 50% of the hair bundles embedded in agarose. And you can also appreciate that the contamination is relatively low if we use nuclei as a measure of contamination because there are relatively few nuclei present. Next slide. And this slide illustrates our basic method that we used, starting again 15 years ago and more or less used for 10 years after that, where we isolated bundles, in some cases separating the proteins by running them on an SDS gel, although we stopped doing that after a while, digesting bundle proteins with trypsin, separating proteins with liquid chromatography, ionizing using electrospray to introduce into the peptides into the mass spectrometer, and then collecting mass spectrums that we could use to search against the database and identify proteins. So that's a basic LC-MS workflow. And over the years, before meeting and collaborating with Ying, we still were able to really improve the sensitivity pretty dramatically by improving sample preparation, getting better LC instruments, improved electrospray with nanospray devices, better and better mass spectrometers. Those of you who have been following the OHSU proteomics core have known that every 3 or 4 years we get a new, better mass spectrometer, and things improve even more. The databases have improved over time. The databases are annotated better, and we have new and improved search algorithms. And altogether, that's meant that, while in 2007 we were able to detect about 75 proteins in hair bundles, in 2013, next slide, please, we were able to detect 650 proteins in hair bundles, all of which, not all of which, but many of which made sense. So in this diagram here, what I'm doing is plotting on the x-axis individual bundle proteins ranked by their abundance. Their abundance is listed on a log scale on the left. And you can see also that each of the bundle proteins is marked by a symbol that is colored based on the different categories in that key. But also the size of the dot corresponds to the enrichment of that protein in hair bundles compared to the same protein in the whole epithelium. And so bundle-specific proteins will have a nice big dot. And what's pretty clear is that of the most abundant proteins, many of them are red, and so that says they're actin enriched or actin cytoskeleton associated, which makes sense given that the stereocilia are filled with actin. And while most of these proteins may be unfamiliar to the audience, the people who work on the inner ear will recognize many of them, because many of them are encoded by genes that are so-called deafness genes. And those are genes that, when mutated, cause deafness in people or mice or what have you. And so it turns out that many deafness genes are expressed in hair bundles, and this is a nice illustration of that. Next slide. And then taking this quantitative information, I haven't talked about how we quantify, but we use label-free methods for quantitation, and then information about where various proteins are located from immunocytochemistry. We're able to develop a model that localizes a fair number of key proteins to very specific locations in the stereocilia, and the stoichiometry and relative abundance compared to other proteins allows us to make hypotheses for what different proteins are actually doing. Next slide. We also switched from chickens to mice. Maybe 10 years ago, we started that switch. And I'm not going to talk about the hair bundle characterization in mice. We did that, and it was quite successful, again, with the vestibular organ. But to set up the rest of the talk, I'll just also point out that we developed techniques to isolate hair cells that were marked they're from a mouse line that labels hair cells specifically with GFP because of a hair cell specific transcription factor. And we were able to use FACS sorting to pool hair cells into pools of as many as 5,000 cells either GFP positive cells, which are hair cells, or GFP negative cells, which are supporting cells of various sorts. And we did that both in the vestibular system, the utricle on the top, and the auditory system in the cochlea. And I'm not going to go through the details here, but I just want to point out that in the data that's shown on the right, we used a couple of different mass spectrometry approaches, data-dependent acquisition in the middle, or data-independent acquisition on the right. And we're able to detect these particular myosins. These are just some examples of proteins we detected. These are proteins encoded by deafness genes that are present at relatively low levels in the bundle. Notice the relative IBAC, the R-IBAC is the fraction of total number of molecules that each protein is present in the sample for. So the myosin 3A and 3B are, well, there isn't an IBAC for 3B, but myosin 3A is 2 parts in a million at a molar level, and yet we're still able to see that protein even with samples of only 50, I believe, hair cells. No, sorry, 300 hair cells is the minimal one. So it's good news, 300 hair cells, but if you're interested in doing things with single cells, that's bad news because we're a long way away from really being able to work with single cells. Next slide. So there are a number of things that we just can't do with the technology we have here at OHSU. So we can't detect the mechanotransduction proteins we're really interested in. We can't, if we were able to pool a few hundred cells, we wouldn't be able to detect thousands of thousands of proteins reliably. And we can't detect proteins from single cells. And the single cells allow us to identify developmental trajectories. We have the advantage of working with organs that allow us to look at developmental trajectories, but we can't do that unless we have single cell sensitivity. And there are a number of other experiments that we're just stuck on. And so we need better technology. And I was very fortunate to meet and connect with folks at PNNL, initially Dick Smith and Ryan, and then eventually with Yang. And that has led to really greatly improved technology for our experiments. And so I will now turn it over to Yang for the next part.
Thank you, Peter. Yeah, I think Peter have make a good point. You need a better technology, but for PNL, we need a better application because we have better technology. We need to know how to better use the technology. So Peter also talked about the great improvement, actually dramatic improvement of sensitivity in the past 20 years for mass spectrometry. Not only the sample prep, ionization efficiency, and LC separation. So PNL have a very long-term interest to improve the sensitivity of mass spectrometry. So this is a paper actually published on 2004. It's the most 20 years ago. It's used a very low flow LC coupled with high resolution FDRCR. It's also similar to Orbitrap, not widely used one. At that time, the Yufeng Shen and Dick Smith tried to evaluate how sensitive it can be. What they do is to inject very low amount, 75 zibtomole of BSA digest into the LC column and try to measure with mass spectrometry. And finally, they realized that we can see all the most of the peptide from the BSA. And if we calculate the detection limit, actually it's very close to single molecular. It's 6,000 molecular. It means if we have 6,000 molecular, we can use mass spec to detect it. So at that time, we know mass spec is not actually not the limit of the is not the limit of the single cell analysis. Next, please. Yeah, so this is just try to conclude what we know about 20 years ago. The analytical sensitivity actually is one cell, because if you look at the copy number of protein in the typical mammalian cells, about over 50% of protein have a copy number over 5,000 copies. So it means analytical sensitivity of mass spectrometry is already can measure over 50% of protein in the cells. But until 10 years ago, we still need 1,000 cells, even million cells to do useful measurement. So what we think at the time is actually the big gap here is sample preparation. So we cannot do very good sample preparation to deliver the sample protein to the mass spectrometry. Next, please. Yeah, this is a very good slide to show how we do the proteomic preparation 10 years ago. So, because we need to extract protein out, we need to first lyse the cell, and we need to transfer the lyse cell into a tube to do protein reduction, alkylation, trypsin digestion. We usually have to do desalting to remove all the band matrix before LC-MS, and then transfer to another, we call it LC vials, as a sample vial to inject the sample. If you look at all this procedure require lots of surface. So if you know the protein is very sticky, no matter all the surface is exposed to, it will stick to it. So if you have very low concentration of the protein, you just lost majority of them before you inject into LC. So it means when you load it into LC vials, it's just nothing. It's everything. If your concentration is very low, it's just lost in all the surface. Next. We start to find a way how to improve the proteomic sample prep. So I joined PNL 6 years ago, and my background is microfluidics and automation. So the idea we have actually is quite simple. We still do the very similar everything we do in like microwave plate. So the only difference is we reduce the volume of the sample prep to nanoliter scale. And as you can see, the chip in the center is a glass slide. We pattern use the saline solution to treat the surface have a hydrophilic spot high on hydrophobic background. So if you put a droplet, it will sit there. So each droplet, what we are doing at the beginning is 200 nanoliter. So, and as you can see, if you look at the video here, we also built a robot because if we use pipetter, it's not enough. It's not precise enough to measuring nanoliter scale solution. So we built a robot in PNL and then do all the sample prep, including everything in one spot, in one droplet. So because we do not transfer them, so we do not have the problem sample loss. So we use the mass-compatible surfactants, so we can do efficient protein extraction and protein digestion. And finally, we transfer them for LC-MS. Next. So here is the comparison between typically tuber method and the nanopod method. See, we reduce the volume almost 1,000 times, and because the volume is smaller, if you look at how much surface the sample exposed to, it's reduced over 99.6%. It means we can achieve about at least 99, more than 100% higher sample recovery. Also, something we also realize is also very important for proteomics is the digestion kinetics, because we need to digest the protein into peptide. If the volume is too large, the concentration of the protein is too low, you cannot get the efficient digestion. So the small volume also help to efficiently digest the protein into peptide. That's why we can measure them in use LC-MS. Also, the throughput is higher because we use a robot. Yeah. Next, please. So here is a kind of demonstration. Use the cultural HeLa cell. So it's just a simple experiment. We try to dilute the HeLa cell suspension from 10 HeLa cell 40 HeLa to 140 HeLa. Here we also show the raw mass spec data. Actually, if you look at the 10 HeLa, the 1E7 is pretty good signal for the mass spec we have. On the right panel, you can see, you can look at the red bar. It can identify more than 3,000 protein from only 10 HeLa cells. If we use the typical way, use the valve at the microliter, like 25 microliter, we only can identify less than 500 protein. So it clearly tells us the small volume is very important to increase the sample recovery, increase the sensitivity to get a much better protein coverage for a very small number of cells. Next. So our first trial to do single cell proteomics is just to coupling flow cytometry to our nanopod device for single cell isolation. At the beginning, we thought it's quite challenging because the wire is so small. But finally, we have very good technician here, so we can able to isolate single cell from the small to the small size in the wires. The wire size here we use is only 1 millimeter in diameter. So we can isolate 1 cell, 0 cell, 10 cell. Because if we use FACS, actually it's good because we can use a fluorescence channel to select only like GFP positive cell or other phenotype selection to select only subpopulation cell for downstream analysis. So we go through all the nanopod procedure to digest the protein to peptide. So here is the first data we get for single cell. We can get 3,000 peptide from 1 HeLa cell, and it's about 600 protein. And if we require at least 2 peptides for quantitation, we can quantify 300 protein. Actually, that at the time is a very good number. We get much better now, but in the beginning in 2018 is a very good number to quantify the protein in single cells. Next. This is the new data we are working on. Is recently published now. Is try to improve the sensitivity of the lab-free single-cell proteomics. We still like lab-free because the it's very clear, it's very precise, and it can direct us the copy number of the protein, and also just sample is much easier. So how we can improve the sensitivity of lab-free approach? Here we use eye mobility. The system we use is a new FAMS source from Thermo Fisher. So what do we do here? We're developing the new data acquisition approach. We call it the TIFF, is transferring identification based on FAMS filtering. So what we do is we first use the spectral library, and it's about 50 cell or 100 cell to generate a spectral library contain each CV, and for each peptide we identify peptide we have the CV value and the retention time and the high resolution MS1 to assign each peptide with these tags. And for single cells, we run through multiple CV. Then we put it into a software, so we can do a kind of match between run, but involve the 3-dimensional match. So the data we show the method is much better for sensitivity. The reason is not the FAMS increase the sensitivity. We can accumulate iron for 10 times more because we fragment iron into different populations. So here I will show later why we can improve the sensitivity. Also, because we use match between run, the 3-dimensional matching also reduces the fault discovery. Next. As you can see, so here is show you why we can improve the sensitivity. If you look at the standard run, it's a black box. So the signal is 2E9, but if you look at the high signal, it's all charged one. And if you run mass spec, you will know they're all from the air or solvent, is not from our sample. So this high abundant contaminant actually take over all your mass detection capability. So that's why we filter out all the charge one species. So if you look at the IT, if you do not filter out this high abundant species, the IT is almost at 20 to 30 milliseconds for each spectrum. After filter out all the contaminated ions, we can increase the injection time from 30 milliseconds to almost 250. It's about 10 times increase in the accumulation time to accumulate more ions for detection. Next. Yeah, here is to show peptide feature is how many peptide feature we detect without FAMT or with FAMT. As you can see, for the 3 different cell line, without the FAMT, we only can see about 18,000. With FAMT, we can see more than 60,000. And also, if you look at peptide, also increase almost double the peptide coverage. Next. Yeah. Here is the data show, to evaluate what's the fault discovery, use the, 3-dimensional matching. If you look at the C and D, so 2D, it means we do not use FAMs. So we have 4% of fault discovery. And if we use 3D, we can reduce to 4% to 1.8%. 50% reduce in fault discovery. Next. Yeah, finally, just we use also HeLa cell because we already use this one to evaluate our sensitivity. As you can see, if we compare the paper published on 2018 and also the recent paper use the TIFF method, we can detect more than 1,000 protein from single cells compared with the older method. So we say double the protein coverage if we use the FAMT detection method. Next. Yeah, it's your turn, Peter.
Okay, thanks, Ying. I just want to summarize that Ying talked about 2 different technologies, both the nanopots sample preparation and then the TIFF method for analysis of the mass spectrometry or introduction of samples into the mass spectrometer. And in early experiments, we didn't have the TIFF method, we just had the nanopot. So I'm going to try and distinguish the experiments in the next few slides with and without the TIFF method. So the questions we started asking initially were, whether we could, with the nanopots, measure proteins in single hair cells from the inner ear. And I'm going to show you some with the chick utricle, the vestibular organ, and the mouse cochlea. And then in the subsequent work, we've been focusing on a somewhat different issue. I mean, given that we have single cell sensitivity, of course, if you analyze more cells, and if those cells are very similar, then you can get much greater sensitivity. And so we've been taking advantage of the sensitivity that Ying has given us to analyze pools of cells as well, single cells. So next slide. So in the first experiments, like I suggested, we used embryonic chicken utricle hair cells. And in the center there, you can see a dissociation from a colleague's lab of these cells. And they and we distinguish hair cells from non-hair cells, the supporting cells, by labeling with the dye FM143, which happens to go in through mechanotransduction channels and specifically label hair cells. Now, just to point out, the hair cells are small compared to HeLa cells, and HeLa cells is, Ying's favorite cell to use as a testing cell. Our cells are about 1/5 the size in volume, and therefore have extremely low levels of protein. Next slide. So on the left, you can see the very simple flow of this particular experiment. We label the hair cells with FM143, dissociate all the cells. You can see examples of dissociated cells there in the middle with FM143 positive and negative cells, and then sort by fax and either put single cells into nanopots wells or add a few cells, anywhere up to 20 cells into a single well. And then in the nanopots chip, we send that to PNNL, and Ying's group does the analysis in LCM-SMS. And on the right, you can see some of these examples. This is from a paper from 2019. So the numbers of identified proteins are relatively small because it was still in early days. But you can see there that we could detect a substantial number of peptides in the FM143 high and FM143 low cells, and that corresponded to detection of a healthy number of proteins, 600 if we had 20 cells, and if we had only single cells, we averaged about 50 or 60 proteins detected. Next slide. This slide shows the proteins, the proteins we detected. And on the left, you can see the results of two independent experiments, each with multiple single cells. Each with multiple samples of 20 cells each. The single cell data is on the right. I'll get to that in a minute. The data on the left are pools of 20 cells, just to illustrate that the reproducibility of detection. If you look at that experiment 1, FM143 high, and compare the different lanes there, if you will, the different single pools, the distribution of intensity, which is coded by the color, is pretty similar from pool to pool, and it's also similar from experiment to experiment. And you can also see that the distribution of proteins is different for the FM143 high and FM143 low. And then a volcano plot there in the middle on the top shows which proteins are highly enriched and are both enriched and then have statistical significance, and a variety of proteins are called out. For the FM143 high proteins, examples like myosin 6, OCM, which is oncomodulin, CALB2, which is calbindin 2. Those are all well-known hair cell proteins. And we were interested in the detection of TMSB4X, which I'll show you a little bit more about in the next slide. But for this period of time now, what you need to know is that TMSB4X is an actin monomer binding protein. And you can see that it is highly enriched in the FM143 negative cells, and then it goes to very low levels. Actually, the quantitation is there in the middle bottom, goes to very low levels in the hair cells, the FM143 positive cells. And we think that's a developmental step. The TMSB4X goes away, releasing a pool of actin, and I'll explain that in the next slide. I'll just point out as well on the right is the back one, please. On the right is that some examples of single cells, and you can see that in experiment 2 didn't work so well, particularly for the FM143 low. We didn't detect very many proteins. Experiment 1 worked better. There's a lot of stochasticity in these data. If we were to repeat this again with the TIFF method, I have no doubt that this would be a far better, we'd see far more proteins. But for 2019, and actually we probably did these experiments in 2018, it was pretty good. So back to TMSB4X. Just to validate this, what we saw on the mass spectrometer, we did immunocytochemistry in the next slide. And in this slide, next please, thank you. We've labeled both with an antibody against otoferlin, which is a hair cell specific protein. And you can see on the top, the cell bodies of the hair cells labeled nicely. And then the antibody against TMSB4X. Now note that in the stroma of this organ, which is the bottom part, which is a lot of connective tissue and various different cell types, the TMSB4X is high. But if you look specifically at the epithelium, where the otoferlin labeling is, you can see that the TMSB4X is found in the cells in between the hair cells, that is the supporting cells. So that validates what we saw with the analysis of the FM143 high and low protein analysis. Next slide. This probably should have been a little bit earlier, but it shows here TMSB4X, which is a tiny little molecule. It binds to actin monomers, and it prevents actin from polymerizing. So the data there in the middle shows that as you increase TMSB4X, the rate of polymerization of actin decreases. And we think this is really important in the inner ear for the reason shown on the right. In the progenitor cells, which are the supporting cells in between, they turn into hair cells during development. We have roughly equimolar amounts of actin and TMSB4X. So there's very little free actin to make any actin structures. But upon differentiation, the TMSB4X levels drop, and we were able to show in independent experiments, single-cell RNA-seq experiments that I don't have time to show today, that's due to transcript levels dropping. And what happens then is, as the TMSB4X is degraded, that releases actin, which can then be used to build the hair bundle, as illustrated on the top. Next slide. We've also, in very preliminary experiments, these aren't published, analyzed one of the two classes of hair cells in the cochlea of the mouse. And in this case, we took advantage of genetic labeling of the hair cells. The FGF8 FLOX GFP will specifically label inner hair cells if we cross those mice with 801 Cre, which is a hair cell specific Cre line. And you can see that just a single line of inner hair cells of the cochlea labeled. And we can dissociate those cells. Now, earlier I talked about the POW4F3 GFP line, and you can see an example of a dissociated cell there, and then the FGF8 cells above that. And we did experiments with nanopots. Again, this is not with the TIFF method. It's the earlier just nanopots, and got better results with the single cells, a deeper coverage of single cells, and pretty good coverage of many cells, many proteins with 7 cells, as you can see there. There was a little more variability though, and looking at those single cells, there are a couple of cells that look dissimilar to the other ones, and I think that's likely because we've probably got some non-hair cells selected in the FACS separation, and so I think more work is going to be required to make sure that our FACS sorting is ideal. But it does illustrate that we can detect a substantial number of proteins in single cells of the mouth from the mouse cochlea, single hair cells. And if we were to scale up to larger numbers of cells, we in principle should be able to detect thousands of proteins, which would allow us to start seeing the relatively low abundance proteins that are in hair bundles. So this is kind of for the future. We have more work to do to improve on this. In the next slide, we shifted gears a bit in the last couple of years. We have another collaborator, David He, who is at Creighton University in Omaha, Nebraska. And David is an expert at isolating individual hair cells and other supporting cells from adult mouse cochlea. And just an example there of the pipette section of a single outer hair cell. You can see it's sitting there in the dish in the left panel, and then it's been sucked into the pipette in the right panel. You can easily do that and suck up a substantial number of cells in an experiment. And by looking through the optical microscope, you can distinguish the two different types of hair cells because they have very different cell bodies. And you can distinguish those from some of the other more differentiated supporting cells that you see in the cochlea. So you can get very clean preparations of isolated hair cells. And you can do things like compare inner and outer hair cells. And that's what I'm going to show you in the next few slides. This first image, this first panel here on the right is RNA-seq data from David's lab, where I've converted their quantitation to relative RPK, which is just the for each transcript, what fraction of the total number of transcripts essentially is from that particular gene. Again, this is transcript data here, and you can see there's a pretty good correlation for most of the proteins. So, this is from about 1,000 cells each in 4 replicates, I think, for each of the cell types. And you can see that there are a variety of proteins in outer hair cells that are enriched, the blue ones, or in inner hair cells that are enriched, the green ones. And we've known about many of these transcripts here, and the genes that are responsible for those transcripts. So this was already published and not terribly surprising. So then David collected lots of hair cells and collected them onto a nanopots chip and sent them to Yang. And in the next slide, you can see the results with the proteomics. And here we've got 500 cells for each of 4 to 6 replicates, 4 for the inner hair cells, 6 for the outer hair cells. And this is again, relative protein expression on a molar basis. So the value there is the fraction of the total number of molecules in the sample. And first of all, many of the proteins that we detected are similar, from the same genes that the enriched transcripts are from. So this looked relatively similar to what the previous slide showed. We could do a volcano plot there on the right and show which of the proteins were reproducibly and statistically significantly enriched. And these are well-known proteins to those of us working on hair cells in the cochlea. For example, oncomodulin is a calcium binding protein, as is calbindin 2, CALB2. Those are known to be specific for outer and inner hair cells. SLC26A5 is a weird transporter that's been turned into a motor protein that allows outer hair cells to be electromotile. That's a whole nother story. That's very clearly enriched in outer hair cells. Otoferlin is a protein that's involved in synaptic release, and the inner hair cells are the cells that have most of the afferent connections to the brain. So all this makes sense. So next slide. So now that we had this protein data, we wanted to do what a number of other investigators have done, which is to compare transcript levels, relative transcript levels to relative protein levels. And these plots are kind of what you often see in these experiments. It's kind of a snot ball, poor correlation, and not really it's either that one of these techniques is not very reproducible or something else. And so we went one more step of the analysis on the next slide. And what we did was we took the relative protein level divided by the relative transcript level and plotted that for inner hair cells versus outer hair cells. And what's fascinating is that brought everything back together again. And we see a really remarkably good correlation between the protein to transcript level in inner hair cells versus outer hair cells. And what that's saying is that any individual transcript, the protein levels from transcript to transcript, the protein levels, it's not always the same abundance as the transcript. It could be much higher or much lower, but that is reproducible from between these two cell types. There's another thing that's kind of interesting in this plot too that we're following up on, which is that the outer hair cell enriched proteins, the ones in blue, fall pretty close to the unity line there, the red dashed line, which suggests to me that most likely they're enriched because their transcript levels were increased which led to their protein levels being increased. So the ratio of protein to transcript didn't change, but the absolute values for each of them did, and that gave the enrichment. It's a little different for inner hair cells. Some of those proteins are close to that unity line, but other ones are way off. I mean, let's just look at PGM2L1. The protein to transcript level is much higher in inner hair cells than in outer hair cells. And to me, a viable hypothesis is that maybe the transcript levels don't change from outer to inner, but maybe the protein is stabilized in inner hair cells, and maybe that's relatively more common in inner hair cells than outer hair cells. So you see more green plots to the right of the unity line. So we're with this added sensitivity and the ability to make these really clean preparations, we're starting to unveil some new biology that we're going to be following up on. Next slide, please. So what's next? We are planning to do experiments where we look at mouse inner hair cells during development. We've done a lot of characterization of their hair bundles during development. I'm not going to go through this slide, but you can see that the lengthening of the stereocilia is different time-wise than the widening. So we want to find out which proteins are associated with lengthening and which are associated with widening. So we're going to pool relatively small numbers of inner hair cells, very specific times during development and at a very specific place in the cochlea, and then look at the protein expression over development. Next slide. And I give it back to Ying.
Peter have shown the data show the disagreement between transcript and protein, and also very exciting to see the ratio actually very consistent. So I think there's a lot of things we don't understand why, and maybe we can provide better technology to do that, because the previous measurement still collects the different cells. So even we know that there are two cell types, but they are not collected from the same mouse, not collected at the same time. So if we can do multi-omics on the same cell, we can get better measurement. So this is actually the very recently preprint published in PNL. So we show we can actually do the parallel measurement of transcriptome and protein from the same single cell. Next. So how we do that? So we call the technology is a nano split. What we do, actually the very simple idea is we sort the cell into a droplet, and to light the cell inside the droplet to release the protein and RNA. Then we align this cell containing array, droplet array with another chip without any cell lysis. It's a PBS. So we just mixing these two droplet array and then split them. So we have kind of divide the cell lysis from one chip to two chip. So after dividing, actually we can use one chip for transcript measurement, another chip for protein measurement. So here is just very early data show we measure 10 cell, 3 cell, and 1 cell. So even for 1 cell, we can measure almost 6,000 genes. Actually, it's half cell. And another half cell we use for proteomics, we can most detect close to 3,000 protein from also half of those cells. It's just the technology we show for further kind of integrate analysis the transcriptome with the proteome to understand more about biology. Yeah, that's the new study I want to show. Yeah, that's all our slides. So any questions?
This is Karen. I always have questions. Peter, the transcriptomic proteomic ratio is a brilliant way of looking at it and the disparity. We have done many of the same RNA to protein concordance analyses in our cancer studies, in tumor and adjacent. And across all tumor types and cell types, we find the same range and snot ball if you look at everything. but then break it down functionally into the types of proteins. We consistently see poor correlation between mRNA and protein and structural proteins, housekeeping proteins, and very tight correlation, what we used to call immediate early genes, transcription factors and signal transduction factors. So I'm wondering, because I'm naive about your model system, whether you who know your genes so well can start to parse out whether some of that may also be true.
Yeah, very good question. We have started asking some of those questions, but not in depth yet. One of the things that did jump out to me when I plotted a particular set of proteins, which are abundant actin-enriched proteins. And now we're talking about hair cells, so it's a little bit different. You wouldn't call them housekeeping genes. What we find with, and this is with the non-muscle myosins and with spectrin and with actin itself and a few others, these structural proteins that are abundant, present in very high levels, they are all clustered, as you might guess, to the high end of protein to transcript ratios. So it's as if they've been stabilized, for example, more than the average protein. That's one interpretation. But we haven't started doing what you're suggesting, and I think that would be really good. Not just plotting, and we have basically two ways of plotting. One, we can plot transcript versus protein to look and see whether we, with some, like for example, do bundle proteins, is the relationship much tighter? And then we have this other plot of protein over transcript versus protein over transcript for different questions. So we haven't done it yet, but we will.
And I don't know whether you have the sensitivity for this yet, but this is a challenge for Ying. You could also look at protein turnover with SILAC type methods.
Yeah.
And that, and that would be informative at the basic science level as well.
Agreed.
That's a lot of work for mouse.
Yeah, I know.
For cultural cell,
so let me follow up with a similar but slightly different question. Peter Ying, excellent model. So I'm looking from my interest from the drug side. Given the sensitivity that you have been able to achieve, and looking at the inner versus outer cell, one of the things which has been a challenge in designing new drugs is the autotoxicity. And for example, the drugs which are in the market, antibiotics, aminoglycoside-based and platinum-based based drugs, autotoxicity is one of the challenge. And whenever we are thinking of designing something new, that always comes up. But the sensitivity has been, given the model that you have, I don't know if you have already considered or not, but I can think of 10 different applications that are relevant to the things that we do. Have you looked at that, the drug toxicities by using this model?
We have not. We haven't really thought about but that's something that would be worth thinking about some more for sure. Because as you say, the ototoxicity is really limiting for these certain classes of drugs. Especially the aminoglycoside antibiotics and the platinum cancer chemotherapy drugs, like carboplatin and cisplatin. And whether that using these techniques will allow us to better screen drugs or what have you, or other methods, I'm not sure. I mean, I think one of the problems has been everything with the inner ear is challenging because it requires a dissection to get an epithelium out, and if your assay is relatively macroscopic, then it's one ear, two samples per mouse or whatever, and screening a bunch of molecules is really difficult. So if we had a single cell assay of some sort, and we were able to dissociate cells into many wells, yeah, absolutely. I have to think about what whether there's a connection with the proteomics or not, but there's no question that you could devise a better screening method with isolated cells.
So just to add a little bit, I didn't mean, considering as you mentioned, not, for example, large number of molecules, but the molecule, for example, we know that cause some sort of autotoxicity. if we can identify one of the challenge, for example, for aminoglycoside-based antibiotics and platinum-based compounds is that to know what is the protein to go after to develop a secondary therapy for that. So if we have treated, untreated, and if we can identify the proteins, that helps with this kind of sensitivity. That would help to then design something new. So not to screen the molecule, but I meant in terms of identifying the proteins given the sensitivity. So I'll let others ask, but there's fun, and we can probably talk offline and more ideas. So
Thanks.
Great. I think we just have one last question from Karsten.
I'm muted.
Great work. It's really amazing what you can analyze with these techniques. So one parameter that should be very interesting is the protein stability. I mean, the lifetime of the proteins. So if you have these hair cells, can you culture them long enough that you can do an RNAi, knock down one of these proteins, and measure 8 hours later or 12 hours later? And if the protein is still there, then it's a very stable protein. And if the protein is gone, then the RNAi worked and there's turnover. So that would be fantastic.
Yeah, we have done some, not that experiment, but some other preliminary turnover experiments. And my former postdoc, Jung-Bum Shim, has done some more detailed turnover experiments. One of the challenges is that with some of these organs, when you dissect them out of the inner ear and put them in culture, they stop protein synthesis for about a day, and then they restart. Now, not every organ does that, but there's some technical challenges with the culturing. I think what we'd have to do is culture for a longer period of time and get into a stable region. I mean, people do culture experiments all the time. They're a little bit tricky because things happen in culture, but they are doable experiments. So the turnover types of experiments are feasible for sure. And Jung-Bum was trying to do turnover on the proteome wide scale, not with SILAC, but with different techniques. And I think that's something that's well worth going back to. And I think, based on our preliminary data, we may want to devise some experiments to test some of these specific hypotheses.
Great, thanks.
All right, well, I want to thank our presenters and questioners. Oh, Fakadu, okay, I'm going to let you have that last question, Fakadu. He wants to know a comment about the cost.
It's not cheap, but Ying kind of can speak to that more.
Yeah, it's not only for us, we also need to know what kind of sample. It's all different workflow. In fact, sorted, if you send out the chip, it's we know how to do that. So if the tissue is another story, tissue we need to use let the capture market dissection also require a lot of time, labor to do that. Yeah, if you have question, you can send me email, so I can find the number for you.
Thanks a lot. I'd like to thank the speakers and all of you and such an interesting thing. And my mind's totally blown by the splitting a cell in half. So anyway, thank you all. And we'll see you in the fall for another P-Medic quarterly seminar.
Okay, bye.
Take care, everyone.